Welcome, and let's get started. We're talking in this video about enzymes, giving an introduction to enzymes, focusing on these few learning outcomes that are listed here. We're going to distinguish between simple enzymes and complex enzymes, describe the two common models of enzyme function, which are the lock and key model and the induced fit model, and then finally learn how to interpret enzyme names. So let's start with the first one, distinguishing between simple enzymes and complex enzymes. Very broadly speaking, we can classify enzymes into those two categories. Simple enzymes, I'll just draw a big blob to represent simple enzymes. In terms of structure, simple enzymes consist of just a big chunk of protein. That's what the enzyme molecule is comprised of, is protein. So it's just protein. So one large protein functions as an enzyme. That's a simple enzyme. Other enzymes, though, are classified as complex enzymes. They might be a similar size, but they are made of two parts. So this is representing a complex enzyme. A complex enzyme is formed of two parts. One part is a big chunk of protein. But the other part is a non-protein part. The non-protein part, what is that? What do I even mean? The non-protein part itself goes by two different names, depending, and I'll show you what I mean depending. The non-protein part might be called a cofactor if it is inorganic. If it is formed of an organic component, it is referred to as a coenzyme. So let me just pause for a moment to make sure. We've just seen a bunch of new names. Simple enzyme and complex enzyme are just the two broad categories of enzymes. The complex enzyme, the structure of the molecule itself is a piece of a protein, but part of that larger structure, there's part of the structure is not protein. So protein and not protein, what do we mean by the not protein part? The name of the not protein part is either cofactor or coenzyme. If it's composed of something that's inorganic, and you know what that means, it means not carbon-based. If it is not carbon-based, then that's called a cofactor. And if this non-protein part is carbon-based, if it is organic, then it's called a coenzyme. So the complex enzyme consists of protein with an inorganic cofactor or protein with an organic coenzyme. And you know what organic means. Inorganic might be metal ions, for example. Here's the cool thing to connect this to something that, I don't know, seems relatable for you. What this means in your body and in your diet is this. The things in your diet that serve as coenzymes are vitamins. Vitamins, as you know, are necessary in your diet. You don't need them in large amounts. And we'll see in a moment that enzymes don't get used up in a reaction. They're not reactants. They're more like a helper molecule. They don't get used up, so we don't need them in large amounts. Interesting. That's part of the reason why you don't need large amounts of vitamins in your diet. Part of what they're used for in your body are to build coenzymes, to build an enzyme, and you don't need large amounts of these in your body in the same way that you need larger amounts of carbohydrate, protein, and so on in your diet. If you think about what else might be in a, say, multivitamin that you might take as a supplement, if you look at the ingredient label on a multivitamin container, you'll see that much of what's in there is vitamins, but pause for a minute and see if you can think what's the second thing I'm going to write. and it is minerals. That's the second thing that you might see in a multivitamin container. Minerals like iron, zinc, and so on. These are things that are necessary in your body. Some of the reason why you need minerals is to be able to build cofactor, to have the structure for a complex enzyme. So it's pretty interesting that things that you know that you need, you maybe never give a lot of thought to why you need those things in your diet and in your body. Part of the reason is to be able to build these complex enzymes. It's not to say that's their only purpose. Iron, for example, is used to build the hemoglobin molecule to carry oxygen throughout your body. That's not the only reason, but this is a pretty big reason for having minerals and vitamins in your diet to build these pieces of a coenzyme. What we've just done is learn how to distinguish between simple enzymes and complex enzymes. The next step for us is to look at the two common models of how enzymes function, how they actually do their work. And before that, or as part of that, we'll say, what is that work? What is it that enzymes need to do? So I'm going to flip the page over to page 214 to start talking a little bit about enzyme function. And as I mentioned just now, I said make sure that we have a sense of what is it that enzymes do, like what is their function. Enzymes speed up reactions. They are not reactants themselves. They are there to speed up the reactions. You've seen that concept before when we talked about catalysts. Enzymes are biological catalysts. Enzymes, I'll say what I just, or write what I just said. Enzymes do not get used up in the way that reactants do. And enzymes, along with speeding up reactions, enzymes often have ability to control the reaction. I'll say something briefly about what I mean by that, control the reaction. You learned when we were talking about organic chemistry, you learned, for example, about Markovnikov's rule, how there's a major product and a minor product when you're hydrating an alkene. You also learned about Zaitsev's rule, how there's a major product and a minor product when you're dehydrating an alcohol. By controlling reaction, what I mean by that is enzymes can steer the reaction toward a preferred set of products. That's probably the most simple way to say that. They help determine what the reaction products will be if there are multiple options. So that's what it is that enzymes do. How do they do it? What is the structure of the enzyme to help us understand the function of the enzyme? There are a couple new terms here, substrate and active site. The substrate is the reactant. This is the reactant that the enzyme will act on. So the substrate is not part of the enzyme. It's a completely separate molecule that comes wandering about and encounters the enzyme. So it's the reactant that the enzyme will act on. Here it's listed in the orange-red. The larger one, the yellow, is the enzyme itself. This is the enzyme itself. What's the active site? The active site is the specific part. This is the specific part of the enzyme molecule. So it's a location as part of the structure of that enzyme molecule. It's the specific part that attaches to the substrate. So the enzyme molecule is this larger molecule with lots of different areas to it as part of its structure. If the substrate molecule comes along, it doesn't click or attach or bond at any random spot in the enzyme. There's a very specific spot where it needs to attach. If that same substrate were to approach the enzyme from this direction, it would sort of just bounce off that enzyme molecule and nothing would happen. But if it bounces off or approaches the enzyme from this direction, then it can attach at the active site. That attachment needs to happen in order for the enzyme to function. The substrate and active site form what's called an active site, substrate active site complex. But our focus right now is just on the concept that each enzyme has an active site for the substrate to attach. Here's a specific example, not so generic as this one. This is showing a model of a specific enzyme called catalase. Catalase is an enzyme that you'll be working with in the enzyme lab that you'll be doing, and it's showing the enzyme itself. This whole thing is the enzyme, and it's showing the substrate. One thing that I would like you to notice right off the bat as I show you this image is to notice this, and it's usually, it's not every single time, but almost always, usually enzymes are huge. Look how big that enzyme is compared to that tiny, tiny substrate. Enzymes are huge compared to the substrate. That's common. And think about it for a moment. What are enzymes made from? They're made from proteins. And you know that proteins are big. Moreover, enzymes are made from proteins often with something else attached to the protein. The cofactor, which often is from a mineral, or the coenzyme, often from a vitamin. Those are large molecules. Here, the active site is highlighted. This is the specific part of the enzyme where that H2O2, that substrate, needs to make its way to that spot. Otherwise, it will not be able to function as an enzyme if the substrate doesn't attach there. So that general overview of structure of an enzyme is going to help us understand enzyme function. We talked on the previous page how there are two types of enzyme function, the lock and key model to explain enzyme function, and then the induced fit model to explain enzyme function. Let's look at this first one. These are the two models. So I'll say models to explain enzyme function. So this is the first of the two models to explain enzyme function. This one is referred to as the lock and key model. And we'll try to show why that makes sense. The purple is the enzyme itself. The yellow is the substrate. The specific spot of that large enzyme that is where the substrate attaches is called the active site, and it's indicated right here and right here. It's showing how, excuse me, it's showing how a substrate approaches. It needs to attach at the active site. Notice the shape of the substrate. This particular substrate has the rectangular thing hanging down next to a triangular thing hanging down. That almost exactly matches the shape of the active site. Here's a rectangular shaped hole and a triangular shaped hole that matches the shape of that substrate quite exactly. So the substrate needs to attach at that spot and in this orientation. It can't have it be facing sideways with the rectangular thing pointing to the side. It needs to be the correct shape and it needs to be in the correct orientation. When that happens, then you have what I called, I won't write it down because you don't need to know this name, but I called it the substrate active site complex. That's where the substrate has exactly attached at that active site. Then what's happening at this time is that the enzyme is enabling the reaction to take place, the reaction that it's helping to control. When the reaction has completed, then the substrate separates from the enzyme and it's now in its products. It looks like this enzyme helped hydrolyze maybe this substrate. There used to be one larger molecule. After the reaction, there are two smaller molecules. Maybe this was a hydrolysis reaction. So lock and key, I'd like you to notice, is very specific. That's probably the hallmark of that model. It's very specific. very much like a lock, say lock to your apartment door, and a key. If I were to shave off the rectangular part of that molecule, it wouldn't make a perfect fit here, and therefore it would not work. So this is very specific. The other type of enzyme function is referred to as induced fit model. Think about what that word induced means, and maybe we'll think about it in the context of something medical. If a woman is pregnant and getting ready to deliver her baby, sometimes the medical doctors need to induce labor. If labor is not already happening on its own, induced labor means let's give it a little bit of a nudge, let's help it along. It needs to be close though. The woman needs to be kind of close, ready to deliver the baby, otherwise it doesn't work to try to induce labor. This is very similar. Induced fit means, well, if the substrate shape and the active site shape are pretty close but not perfect, let's just give it a little bit of a nudge, and we can sort of wiggle it around and make it work. That's what induced fit is. Check it out. Of course, that exact shape of the substrate doesn't exactly fit the active site shape, but it's close. There's two kind of holes or indentations where the rectangular and triangular points or parts of that substrate could fit. It needs to be close. It wouldn't work over here, but that's fairly close. It's not perfect, so it's not a lock and key type of model, but it's close. So the substrate comes in, and the interactions between, intermolecular attractions between the substrate and the enzyme allow the enzyme to kind of adapt shape a little bit to attach the substrate, for the substrate to attach. So you still get the same type of active site complex. The difference is that it's not a perfect fit. It's a sort of a close, good enough, will make it work fit. That's what induced fit model is. And again, you have products after the reactions has happened. So this is less specific. I won't say not specific at all, and we'll see examples on this next page what I mean by less specific. There's still some specificity involved, but doesn't have to be as exact. These are the two types of models that biochemists have come up with to try to explain how enzymes function. So we have just now talked about the two common models of enzyme function, lock and key and induced fit. The last thing we'll do in this video is learn how to interpret enzyme names. Enzyme names are pretty cool. They let you know what is the substrate, what's the reaction type, what kind of model are we talking about in terms of how the function works. All of that is included essentially in the enzyme name. And so let's take a look at that. Let's look at how enzyme names are given. First thing that I'll ask you to notice right off the bat is that the names, these are the names. I guess I didn't write that. These are the enzyme names in this first column. First thing I'd like you to notice is look at the ending. That's often how we've done the naming in this course, is we start by looking at the ending to see what kind of clue that gives us. ACE, 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 and so on. So I'm going to ask you to notice enzyme names usually end in ACE. Enzyme names usually end in, enzyme names usually end in ACE. Not always. Let me slide the page up for just a moment. I've got a few on the bottom. I put those in italics because they clearly are in a different category. We're not going to analyze their names, but I listed them because one of them you've worked with in lab. This is from the milk lab, the lipid lab, pancreatin. That's an enzyme formed in the pancreas. What I'd like you to notice about these three enzymes, their names end in IN. These are kind of some old-timey enzymes. These were some of the enzymes that were first identified and isolated by biochemists. They are found in the digestive system. That should make sense if you were a biochemist in the early years trying to make some sense of biochemistry in the body. Digestive enzymes are easy to get at compared to other enzymes that might be inside cells throughout the body. These are in the digestive juices and so on. And so these were easy to isolate and identify. They named them with the IN ending. And those are kind of more common names, you could think of it. The ones that end in ACE are the much more systematic and modern, I guess you could say, names. So those are the ones that we will be learning how to name and interpret the names. You won't be interpreting the names of these. They don't follow the systematic naming pattern, but I do want you to recognize that some enzyme names stand alone. They don't follow that pattern. Let's look at that pattern. I wrote the enzyme name. We first said, oh, look, it ends in ACE. That's common. The second thing that I would like you to notice that all of the enzyme names have is this. The very first word listed tells you what the substrate is. That's cool. That tells you that this enzyme, glucose oxidase, will be acting on the substrate glucose. Remember what substrate is. That's the reactant molecule that the enzyme is going to help react. So the reactant molecule, the substrate, is glucose. I see that there's a second word. The second word gives you an indication of what is the reaction type. So let's fill in this table that I have written for you. The substrate for glucose oxidase would be glucose. Take a wild guess at what the reaction type is. If it's indicated by the word oxidase, take a guess. And I think you are right. The reaction type is oxidation. What does that tell you about enzyme model? Remember that the enzyme model will either be lock and key or it will be induced fit. And remember what the difference is. Lock and key means it's very specific. Induced fit means it's less specific. So in the context of the naming, what that means is if the substrate is a specific molecule, not a category of molecule or a general classification, but a very specific molecule, then that is specific. And so that's glucose, right? Glucose is not a category, it's a specific molecule. And specific then indicates that that enzyme will follow the lock and key model of enzyme function. Let's take a look at the next one. What is the substrate? It would be the first word, cytochrome. You don't know that word necessarily from this class anyway, but you still can interpret this enzyme name based on the pattern we've just identified. The reaction type, oxidase, so it's still oxidation. Cytochrome refers to something very specific, and if it's specific, then that is a lock and key function that's being followed. How about this example? Sucrase is an enzyme that you have worked with in the lab. That is one when you spit into a test tube, trying to hydrolyze sucrose. I see what the first word is. The first word, remember, is always the substrate. I didn't write that up here, but we could. So recall that the first word listed is the substrate. The second word listed indicates the reaction type. First word, second word, and so on. First word is substrate. Notice, though, in this example, it's empty, right? There is no second word. Here's what to do about that. If this word is missing, in other words, if there is no second word, it means that the reaction type is hydrolysis. That's a common reaction type. We've been seeing this bit by bit over the last few weeks. When you eat something, put it in your mouth, chew it up, swallow it, or drink it, it's often something big, right? Like a polysaccharide or a triacylglycerol, or as we've just learned about proteins. Those bigger things, before your body can do much of anything with them, they need to get smaller. And you know that that's what hydrolysis does. It's like scissors. It takes something bigger and makes it smaller. That's a common enough thing that that we will see a fair number of enzymes that the reaction type is hydrolysis. Why it's not called sucrase hydrolase or hydrolysase, I don't know. I wasn't in charge of this naming and neither were you, but we can at least identify the pattern. If the word is missing, that means it's hydrolysis. So this is hydrolysis. What I do want you to notice, though, is that first word indicates the substrate. It's not necessarily the exact word. Like sucrase itself is not the substrate. Sucrase is the name of the enzyme. So you do need to pause and ask yourself, what is the actual molecule that will undergo hydrolysis? It's not sucrase, it's sucrose. Sucrose is the sugar, and when sucrose undergoes hydrolysis, the enzyme to help that happen is sucrase. So you just pause for a moment and say, wait a minute, is it actually that word itself, or is that word kind of a hint of what the word is? Sucrase is the enzyme, so what will actually be reacting? Sucrose. Sucrose is a specific molecule, so this again follows the lock and key enzyme, or the lock and key model. How about this one, methemoglobin reductase? Methemoglobin is the substrate, hemoglobin. The reaction type appears to be reduction. This is a specific molecule, so this is lock and key once again. Let's do two more. Actually, we can do all three. Lipase, protease, it's missing the second word, so you know what that means. That means that the reaction type is hydrolysis for both of these. What about the substrate? Look at the first word and say, what is it that's going to be undergoing hydrolysis in this for this enzyme? Lipase is the enzyme. What does it act on? It gives an indication. It's not perfect. You have to kind of think about it for a minute, but sucrase acts on sucrose, lipase acts on lipids. Lipase acts on lipids, helps lipids undergo hydrolysis. When you identify lipids as the substrate, that should help you realize that this will follow the induced fit model, because lipids is not a specific, that word lipids is not the name of a specific compound, it's the name of a general category. And a general category tells you that it's induced fit. And here's where I was talking about on the previous page. This model is less specific, but that doesn't mean it's not specific at all. This will work to hydrolyze lipids. It won't work to hydrolyze carbohydrates or proteins. It'll work to hydrolyze lipids, the category, but within that category, it's induced fit. It can work on any number of lipids. Protease does hydrolysis on, you guessed it, proteins. And again, proteins are a general category, that's not a specific molecule, that name. This is the name for a general category. So these also follow the induced fit model. And the very last one I want to do with you is this one, because it's a chance for me to sneak in just a little bit of a review. It looks like the enzyme name is lactate. I'm sorry, the substrate name is lactate. How about the reaction type though? I want you to remember that remember the prefix D, like de-isoplane means get rid of the ice. Dehydrogenase means remove hydrogen, which is lose hydrogen. That's what dehydrogenate means. And this is the review I want to remind you of. You already know that oxidation in general, that reaction type is gaining oxygen. If you gain oxygen, you lose something, hydrogen. Reduction, of course, is losing oxygen, gaining hydrogen. This general summary is something that we'll be revisiting again in these next days. And so you say, wait, what kind of reaction is it when you're losing H, you're removing H? That's oxidation. So it's a little bit, might seem odd. You might say, wait, why don't they just call it lactate oxidase? It's because the lactate is not actually gaining O, it's losing H. Those are two different ways that oxidation can happen. And so this name is indicating it's oxidation because it's that molecule's losing H. But when you recognize what oxidation is, then you can recognize this as an oxidation reaction. Lactate is a very specific molecule, not a category, and since it's the name of a specific molecule, that means that lactate dehydrogenase follows the lock and key model of enzyme function. So I've just given you a number of examples of how to interpret enzyme names by identifying the substrate, identifying the reaction type, and then identifying what model of enzyme function does that enzyme follow. Where did my page just go? Right here. So what we have, hang on, I've lost my, here we go. How we started was identifying the three topics or objectives we wanted to address in this video, and we've just finished interpreting enzyme names. And so that wraps up our video this time. The next video we'll be talking about how do enzymes help with metabolism. Like, what's the role of enzymes in metabolism, actually helping to get the energy from the food we eat. That'll be for next one. I hope you join me then. Thank you very much, and we'll see you next time.